Hello, everyone. I left a question in last lecture and asked you virtually produce recombinant AR protein using LNCap cells. You will use several technologies we talked about in last lecture. First, in order to produce a recombinant protein, I wonder what do you need? We learned how to produce insulin last time, right? So both AR and insulin are proteins. Therefore, these recombinant proteins could be produced in the same way. Then we know that we need AR cDNA to generate a plasmid DNA that can express AR. Then where can you get the AR cDNA? We learned that cDNA can be made from mRNA by reverse transcription or RT, right? So that means if we can get mRNA of AR, we can solve this question. So this question tells us that we have the LNCap cells that can express AR, suggesting that we can extract mRNA from the LNCap cells. Okay, so based on this logic, you now have this answer. The flowchart shows these steps. First, okay, you can obtain mRNA from the LNCap cells, and then cDNA can be made by reverse transcription. So during these two steps, the mRNA and the cDNA of AR are combined with all other mRNA or cDNA. That's the total mRNA or cDNA inside the cells. But during the next step here, the PCR step, you can use the sequence-specific primers to amplify AR cDNA only, but not any other cDNAs from LNCap cells or LNCap cells, okay? So then by restriction enzyme digestion here, you can generate AR expression plasmid, which can produce AR protein, and then this is in bacteria. The final step, of course, will be purification of recombinant AR protein from the bacteria. I hope you got this answer correctly. Okay, today we are continuing the same topic. I will introduce more genetic technologies to you. In the practice question, we discussed how we can generate the recombinant protein using genetic methods. But actually, many recombinant proteins are being used therapeutically. For example, the growth hormone is a peptide hormone secreted by the pituitary gland that stimulates growth and cell reproduction. It is prescribed when children have growth hormone deficiency. In the past, growth hormone was extracted from human pituitary glands. Similarly, other therapeutic proteins were obtained from slaughterhouses and donated human blood. But with recombinant DNA technology, these therapeutic proteins can all be made safely and efficiently in vitro. Okay, we can use bacteria, genetically modified animals, and plants to make these proteins. For example, the blood clotting factors and the insulin, they are made in bacteria in a large scale. Human insulin was the first human protein made using recombinant DNA technology. Currently, we still do not have a curable therapy for type 1 diabetes. So some patients with type 1 diabetes must inject themselves with insulin several times a day to control the blood sugar. So this table lists some products made by recombinant DNA technology and used routinely in clinics to treat patients. I'm not going to go through them in details one by one, okay? But you just remember that there are recombinant proteins that can be used in clinics. Although bacteria or sometimes yeast or plants can produce large amount of recombinant proteins, many therapeutic human proteins are produced in mammalian cells. okay? Most of the time, the recombinant proteins produced in mammalian cells have the correct modifications and folding. Therefore, the protein activities are preserved much better. The mammalian cells can be grown in large batches in those equipment called bioreactors shown here. One of the proteins produced in bioreactors is named myozyme. Okay, the myozyme is used to treat this Pompe disease. Pompe disease is also called a glycogen storage disease type 2. It is autosomal recessive metabolic disorder which damages muscle and nerve cells throughout the body. It is caused by an accumulation of glycogen in the lysosomes due to deficiency of lysosomal acid alpha glucosidase enzyme. Okay, so this drug, Myozyme, is very expensive. The average cost for Myozyme is about $300,000 per year and ranked one of the costliest drugs per patient. Okay, now we'll switch the gears and talk about the stem cells and how we can use the stem cells for therapy. The most important feature of stem cell is its ability to differentiate into different cell types. Therefore, stem cell itself is unspecialized, meaning undifferentiated. Okay, as the undifferentiated cells, stem cells can proliferate by cell division. We call this process as self-renewal. Okay, during the stem cell self-renewal, it still maintains the ability to differentiate it into other types of cells. This is the reason when we talked about embryo splitting last time, when the early embryo cells named blastomeres are separated into individual cells, each cell can develop into a full animal, right? So blastomeres can further develop into blastocysts. Then we learned, we tested that before, right? The inner cell mass of blastocysts are stem cells, and we call these stem cells as embryonic stem cells or ES cells. The later ES cells were developed into an embryo. So ES cells are pluripotent, meaning that they have the ability to differentiate into all kinds of cell types. This is important. It's going to be tested later, okay? In addition to ES cells, stem cells can also be found in adults' body. And we call these cells as adult stem cells or somatic stem cells. For example, hematopoietic stem cells in the bone marrow and umbilical cord blood can differentiate into all blood cells. Stem cells in skin can self-renew and differentiate into different cell lineages of the skin. Okay, adult stem cells are multipotent, meaning they can only differentiate into some or certain cell types, but not all, okay? So we previously learned that cord blood cells have been approved by FDA to treat more than 80 different diseases, and this is due to their multipotency, okay? This slide shows how ES cells can be isolated and cultured in vitro, okay? We have learned the blastocyst, which is a later stage of development than the blastomere. So we have tested that the inner cell mass in blastocyst can further develop into the fetus, right? So in this figure shows a human blastocyst with the inner cell mass under the microscope. The inner cell mass is the dense cell population shown here. Okay, cells from this region are pluripotent and will form all the 200 cell types in our body. Okay, in the lab, cells from the inner cell mass can be removed. and cultured in the petri dish, in the incubators. And then they will grow into or form this embryonic stem cell lines, okay, cell lines. So this is a picture of what ESL or embryonic stem cells look like in cultured plate under the microscope. Due to its self-renewal feature, these ES cells proliferate very fast in vitro, okay? If they can be maintained at the undifferentiated stage, they can live almost forever. Their culture conditions are very stringent. So if you don't use the appropriate culture media, or you don't change the media on time, the ES cells can quickly differentiate. And here shows a picture that the differentiated ES cell look like, okay? On the top, these ball-shaped cells are a cluster of ES cells, and the cells at the bottom, those greenish cells, are most likely feeder cells, okay? The feeder cells are irradiated cells that do not proliferate, they don't divide, but they secrete substances that can help the ES cell to grow. Okay, in this picture, all the cells have been stained with fluorescent dyes, so you can see all the cell structures much better. You may notice that some of the ES cells on the edge, on this side, okay, they started to differentiate into neuronal type of cells with this long extended axons formed. But why scientists are interested in stem cell research in vitro? Because stem cells have many medical uses. The stem cell-based therapies are based on differentiation of stem cells into specific cell types for transplant into patients, okay? The source of stem cells can be embryonic stem cells or adult stem cells. Because ES cells are pluripotent and can differentiate into any types of cells, making these cells the best candidate for therapy. Diseases that could potentially be treated by pluripotent stem cells include a number of blood and immune system related genetic diseases, cancers, Parkinson's disease, blindness, and even spinal cord injuries. However, ES cell therapy involves ethical concerns. It is controversial, right? So additionally, there's a technical problem of immune rejection, meaning a transplant recipient's immune system attacks the transplanted organ or tissue. So another source of stem cells we talk about is adult stem cell. So adult stem cell is multipotent, so it can only differentiate into certain specialized cell types, right? So if the adult stem cells are taken from this patient, there will be no immune rejection of transplanted cells and no ethical concerns, right? So the examples of applications were like bone marrow transplantations, infusion of differentiated heart cells, and burn treatment. This slide shows one application of adult stem cell therapy. The stem cells are removed from the bone marrow of a donor, grow in the laboratory, and then infused into patients with leukemia who had their stem cells destroyed by radiation. Because leukemia is a disease caused by overproduction of white blood cells by stem cells in the bone marrow, after radiation, all the cancer cells will be killed, theoretically, okay? And then the transplanted stem cells will migrate into the bone marrow and begin production of normal blood cells there. That's for the therapy, how it works. Then this slide shows another example of adult stem cell therapy. Basal stem cells can be removed from a small region of skin cells, amplified in vitro, and then they are spread over burn areas, where the stem cells reform skin, bypassing the need for very painful skin grafts. So that's how stem cell can help to treat the burns. Okay, we talk a lot about stem cell and differentiation, right? Now I would like to discuss the process of differentiation. Okay, first, what is a cellular differentiation? So cellular differentiation is the process in which a cell changes from one cell type to another, okay? Usually the cell changes to a more specialized type, showing here, like a stem cell can be differentiated into tissue-specific cells, okay, such as the red blood cells, hematopoietic cells, muscle cells, and others. The environment the stem cells stay determines the cell types that the stem cells were differentiated into, okay? The question here is cell differentiation a reversible event? As early as 1957, a person called Conrad Wallington, so he's a British biologist, described that mammalian development is unidirectional. So that means the embryonic stem cell developed into a more mature differentiated state, but not the other way around, okay? So this idea has been depicted as a Wallington epigenetic landscape shown here. So the Wallington links the process of cellular differentiation to a marble, okay, which represents a pluripotent cell on the top of a hill. The hill contains many paths or the ways that the marble can roll down, and each path will eventually lead to a distinct final differentiated state, such as like a blood cell or a neuron. As the marble moves down the hill, the path and the final destination available become more limited, representing the increased differentiation of the cell, okay? So when a cell moves toward its final differentiated state, represented by the movement down the hill along an increased restriction path, its potential to become anything diminished. So in addition, mutations which alter the way down the slope will have an impact on differentiation of the cell. But does this direction, the unidirection, still hold true? Or is this need to be revised after so many years of development of genetics, this knowledge here? Recent science advancement prove that the cell differentiation actually can be reversed. Induced pluripotent stem cells, also known as iPS cells, okay, are a type of pluripotent stem cells that can be generated directly from a fully differentiated cells, okay? A iPS cell can be created by transfer of certain genes into the nucleus of a somatic cell. The process of generating iPS cell is called cell reprogramming, okay, also reprogramming. So do you all know that genes are involved in reprogramming? What kind of genes are involved? We learned that actually. So these are transcription factors, the drivers for transcription. Okay, let's watch a video of iPS cells next.
Hi there. Did you know that about 1 in 12 people in the world suffer from diabetes? Out of these, 10% are type 1 diabetics, meaning the insulin-producing cells in their pancreas, the beta cells, are defective. Could we culture and transplant into these people beta cells able to replace the diseased ones? First, we need to understand how cells arise in the body. When sperm and egg fuse, they give rise to the zygote, which in turn divides multiple times to form the embryo. Cells in the early embryo have the potential to originate any of the more than 200 different cell types found in the human body. To this characteristic, we call pluripotency. Cells then specialize, acquire functional maturity, and lose potential during a process termed differentiation. Why are cells in the same organism so distinct, even though all share the same genes? A complex mix of internal and external clues determines cell fate, activating and deactivating certain genes. In highly differentiated cells, parts of the genome are permanently blocked. Waddington illustrated this concept in his landscape. Just as marbles can travel uphill after falling from the top of a mountain, mature cells don't usually revert their specialization. But could they? Shinya Yamanaka decided to tackle this question and try to obtain stem cells from specialized cells. To achieve this, he first inserted a set of 24 genes associated with pluripotency into mice fibroblasts. They started to change shape and became very similar to embryonic stem cells. Then, Yamanaka repeated the experiment, reducing the number of genes and trying different combinations. His conclusions were surprising. 4 of the genes were enough to reverse fibroblast specialization. The cells obtained were called induced pluripotent stem cells. These could later be induced to differentiate into other cell types, such as nerve or muscle cells. In 2012, just 6 years after publishing his results, Yamanaka received a Nobel Prize. His discovery has endless applications in medicine. iPS cells can be used to culture defective cells in vitro, providing a tool to study disease development and drug effectiveness. They may replace embryonic stem cells in research and therapy, thus overcoming various ethical issues. And they could help type 1 diabetes patients by providing them with new healthy beta cells. It's an open field of research and there's a lot to discover. Will you join?
Well done, man. High five.
Since iPS cells can be divided directly from adult tissues, they not only bypass the need for embryos, but can be made in a patient-matched manner, which means that each individual could have their own pluripotent stem cell line. These unlimited supplies of stem cells could be used to generate a transplant without the risk of immune rejection. Okay, showing this picture, the iPS cells differentiate into various types of cells. Say if the patient has diabetes, then beta cells can be generated and replace the dead pancreatic islets. If the patient has liver failure, for instance, and then the hepatocytes can be generated from iPS cells and replace the damaged hepatocytes. These types of treatment is named as regenerative medicine. Currently, scientists work hard on figuring out the best conditions to induce the differentiation of iPS cells in vivo. But then, on the other hand, iPS cells can be differentiated this in vitro and used for drug tests in order to develop a new therapeutic drug application. They are very useful research tools. Now we will discuss another technology named DNA profiling. Okay, DNA profiling is the analysis of DNA samples to determine if they came from the same individual. It has another name as DNA fingerprints. Since its introduction in 1986, DNA profiling has become a standard tool of forensics. The basis of this technology is to find small differences among people's DNA. These differences are presented as variations in the length of certain DNA sequences called mini-satellites. Okay. Mini-satellites are nucleotide sequences about 14 to 100 base pairs long, organized into clusters of varying lengths on different chromosomes. The specific DNA sequences used for DNA profiling are called short tandem repeats, or STR. Okay, these are short repeated sequences that are 2 to 9 base pairs long, and in different people, the numbers of these repeats at each STR locus are variable. Okay, this allows the law enforcement and government agencies to identify different people. Actually, now in the United States, a standardized set of 13 STRs, also called a CODIS or CODIS panel, that means combined DNA index system, that is used for DNA profilings. Short tandem repeats, or STRs, consist of short sequences of nucleotides that are repeated many times tandemly. Okay, this is an illustration to show how STR look like. These are the 2 DNA samples. One was collected from the crime scene, and the other one was collected from a suspect. Let's see if the suspect is the bad guy. Okay, analysis on 2 different STR sites was done. So in STR site 1, the STR sequence is AGAT, and the number of STR repeat are the same between the DNA samples, these 2 DNA samples from the crime and the suspect DNA. Okay, so they both have 7 repeats. However, on the other STR locus, the STR sequence is GATA. The numbers of STR repeats are very different. Okay, crime scene DNA has 8 repeats, whereas the suspect have 13 repeats. So based on this result, we conclude that these 2 DNA samples do not match, and the suspect is actually innocent. Okay, this slide shows how STR profiling is performed in labs. First, you need biological samples that contain DNA, right? So like cell showing here. In this case, the DNA extracted is the genomic DNA. And then a multiplex PCR is performed to amplify those STR alleles. Multiplex PCR is a modified version of PCR, meaning that in one single PCR reaction, several STR alleles can be amplified at the same time. Okay, so instead of obtaining one single PCR product from a traditional PCR reaction, multiplex TCR will generate multiple PCR like products. Okay, then here shows many of them. After PCR, the amplified DNA can be separated by electrophoresis and then subjected to result analysis and interpretation. So then you will be able to find out how the results are. So we learned that gel electrophoresis. We'll also learn the PCR. So you will be able to understand the whole process. Let's take a look at the results together. The pedigree shows a family with like a mom and a dad and 5 children here. This is an example, okay? So you can see that each of the bands in the children's DNA from here, each band showing here, you can, they are represented in the parents, and any of the bands showing here, like this one, you have this in the dad, and then this one you have a band in the mother. This is understandable because all the children's DNA all come from their parents, right? However, each child, they have their unique overall pattern of this PCR result, right? So with this technology, it becomes very easy to confirm the relationships between family members. So we probably will have similar, PCR results or patterns showing you during the exam, and we'll see how you can identify the relationships between every individuals. Okay, so this is a chromosome localization of the CODIS panel the police actually using right now. So I'm not going to go to the details, but you can see all these different SDR loci. They are located on the different regions in our genome. So then some of you may ask, is this a reliable technology? This is a calculating probability in the DNA profiles. For example, this CSF1PO has 25 different patterns in population, and the probability of matching one specific pattern is 1 out of 25. Similarly, the STR TPOX has 100 different patterns in all population. Therefore, the probability matching one specific pattern is 1 out of 100, all right? So in this case, the combined frequency of a person matching specific patterns of both CSF1PO and STR TPOX is 1 in 2,500. Okay, so based on this calculation, the likelihood that any two individuals, of course, this is except for the identical twins who has exactly the same genetics, we know that, right? So just two unrelated individuals will have the same 13 loci DNA profiling. They are the same, can be as high as 1 in several billions or even greater, okay? Therefore, DNA profiling is a highly sensitive and accurate technology for genetic determination. Because of its accuracy, DNA profiling can be used as tools for identification of forensic and paternity cases, studies of human evolution and ancient migration routes, and even monitoring food contamination. Okay, the DNA samples can be prepared from many possible sources, including like single hairs, licked envelope flaps, toothbrushes, cigarette butts, and even from mummies more than 2,400 years old. But how comes DNA can be preserved for so long? One reason is that the enzymes that degrades and destroy the DNA is named the DNAase, okay? So this enzyme is very unstable and easily lose activities in air or warm temperature. Also, DNA have double-stranded, so structurally, DNA is much more stable than RNA, okay? But what happened to RNA? But in contrast to DNA, RNA is very unstable. So RNA probably cannot survive for 1 day at room temperature. They will be destroyed. The reason is RNA is degraded by an enzyme named the RNase, okay? RNase is one of the most stable protein and can be found almost anywhere, including on your hands. You will have RNase. Okay. So this part is a little bit confusing. Please make sure that you understand these relationships. Okay. So because of these reasons, if you work in a research lab, always remember to put your RNA samples on ice when you are using those samples, and store the RNA sample in the -80 freezers as soon as you complete the experiment, okay? So therefore, you can imagine that for those samples sitting there for a long time, what we are using for DNA profiling is using the DNA as sample, never use as RNA, right? Okay, so let's take a look at a DNA profile prepared for a criminal case of sexual assault, okay? One short tandem repeat was amplified by PCR from evidence that included criminal's semen and the victim's cells. Okay, bands from these samples were compared with bands generating from DNA samples from the victim. So, and then also her boyfriend, and two suspect 1 and 2. So can you tell who is the bad guy here? So you can pause here, take a look. But then what the answer is, the suspect 1 is the bad guy, because the DNA profile has the same pattern, the suspect 1, as the semen showing here. Okay, so now you guys can all be detectives. In our future exam, you will be asked to solve cases using DNA profiling, right? Now, DNA databases are becoming important tools in solving crimes. Since 1998, the FBI began cataloging DNA profiles in a database now called the National DNA Index System, which collects profiles submitted by state and local criminal justice agencies. In Virginia, which has one of the oldest and largest DNA databases in the United States, more than 3,600 crimes without suspects were solved over a 6-year period by matching DNA profiles from the crime scene with profiles already in the state database. So it's a very powerful tool. So this is a this is not going to be in the exam just for fun for you to know. This is a real case of the it's called the Golden State Killer. Okay, the masked gunman who terrorized California in the 70s and 80s with a series of killing, rapes, and assaults. In 2018, DNA led to this person, this bad guy, D'Angino's arrest. Okay, authorities used a DNA database called a GED match to try to track down the killer who had invaded them for decades. They created a profile with crime scene DNA, and in April 2018, D'Angino's name emerged in what investigators believed was a pool of possible suspects. Okay, detectives then gathered D'Angino's DNA. Some of it taken from the handles of his car door, and some from a discarded tissue in his trash. And then the police found they had a match to the evidence. The left picture showing here, this was a depiction from the witness. And this picture is what the D'Angino when he was arrested. Okay, that's very interesting. Okay, that's all for today.